I'm Kim Samko. I am here today to introduce both a collaborator and a friend, Summer Gibbs, who is, let me just to make sure I get this right, the Douglas Strain Endowed Professor of Biomedical Engineering at Oregon Health and Science University. A lot of us in the audience today know Summer from collaborations, and also I met Summer myself here 17 years ago in her last year of grad studies and my first year as a postdoc. So I've been lucky to collaborate with her since. Today she's going to tell us a little bit about the clinical translation of a fluorescent nerve agent that she's been developing, or a series of nerve agents, I should say, she's been developing. And it's been really nice to have a colleague that is also going through the trials of getting through clinical translation together. And then we're also going to talk, she's also going to give a little talk about some of the intracellular imaging agents that she's been developing for quantitative imaging. So I'm just going to hand over the floor. And also just to note, Summer does have a startup company called Trace Biosciences that is translating the nerve fluorophore. So hopefully we'll hear a little bit about that.
Awesome. Great. Great. So thanks, Kim, for the introduction, and thanks for the invitation. It's really nice to be back visiting. I think I haven't been here since I moved to the West Coast, like 12 years ago, so it's really amazing to be back here and to see all the changes and not that have occurred on campus. All right, so I'm going to tell you today about the technology development we've been doing in my group. We might have to turn the front lights down. I don't know, we can play around with it. My group makes near-infrared contrast agents, and we're looking at ways that this might help us to improve clinical medicine. So as Kim said, here's my disclosure slide. So I'm the co-founder of our startup company. It's interested in our nerve contrast agent development that I'll talk to you about. So my lab does a variety of imaging technology development projects, but they really fit kind of into two umbrella spaces, and I want to take a little time to tell you about each of the technology development projects that we're doing in our group. So I'll start off and talk to you about the nerve contrast agent development and how we think we could use this to improve surgery using fluorescence guidance. So I will share with you a little bit about how the medicinal chemistry project that we've been doing and where we are kind of in the clinical translation pipeline. And then I'll spend the latter half of the talk telling you about this technology that we've been actually working on in collaboration with Kim and Dr. Kenneth Tischauer, who maybe some of you remember was a postdoc here at Dartmouth. And so we've been thinking about developing fluorescently labeled drugs to be able to look at drug distribution and coupling this with cyclic immunostaining to be able to evaluate the spatial proteome and how we might use this technology to look at personalized medicine, treatment response, and treatment resistance. Okay, so I'm going to start here and just kind of give a broad general introduction to the idea of fluorescence-guided surgery. I think probably many of you might have familiarity with this, but what I'm talking about is this idea of sort of helping to improve surgical navigation by adding exogenous fluorescent contrast. So you can think of this as like cut by color or like paint by number. So I'm showing you two of the clinical imaging systems that have the sort of highest use space and implementation in the operating room. So here you can see the Stryker SPY system and then also the Da Vinci surgical robot. So both of these systems exist in the clinic, and there are a number of imaging systems that are already out there. In fact, both of these manufacturers sell their imaging system with the imaging, sell their surgical system with the imaging channel on board. And so I would say that myself and other people in the field are really excited about this technology, but in order to really wield its power, we really need targeted contrast agents that highlight the tissues that are diseased and the tissues that are potentially normal that we'd like to avoid. So I actually learned this morning that this list is slightly out of date, but I will tell you about some of the clinically approved fluorophores that currently exist. So right now, for a long time, there's been fluorescein and the aminolevulinic acid protoporphyrin IX system that I think many people here at Dartmouth have worked on. So these are primarily used in the neurosurgical space to help with highlighting various pathologies. They're not near-infrared contrast agents, and I'll talk to you in a minute about why this is important. And then there's methylene blue and indocyanine green. So these are near-infrared contrast agents, but they're not really targeted to anything. Generally, they're just blood pool agents, and so you're able to look at perfusion and vascularity. Because they are FDA approved, it's been discovered that they do highlight some pathologies. So, for example, methylene blue highlights insulinoma in the pancreas, and indocyanine green highlights liver cancer and liver metastases. So that's sort of a discovery because of the ability to look in many patients. And then, excitingly for the field, the first molecularly targeted contrast agent, Cytolux, or OTL38, which was developed by On Target Laboratories, a startup company out of Purdue, was really recently approved by the FDA after phase 3 clinical trials. So they received approval for ovarian cancer resection in December of 2021, and then for lung cancer resection in December of 2022. And actually, I don't have a slide about it here, but just last week, there was actually the next approval of a targeted contrast agent. So LUM-15 was recently approved by the FDA for breast cancer resection. So definitely there's starting to be some traction in this space and thinking about molecular targeting of specific structures. So how can this help? So here, this is an example of the first human study using the Cytolux type molecule. So you're looking at ovarian cancer. Potentially you can see some of the abnormal structures here, but if you use the fluorescence image, you can see that you're a lot better able to visualize all of the cancer in the peritoneum. And so you can imagine how this might help to have a greater resection than with the white light image alone. So the other thing I already kind of mentioned is that the other idea that I want to put forward is that we want to image in the near infrared. So the reason behind this is that the near infrared window is thought to be the biologically, optically clear window. And this is really because of the tissue optical properties. So we're going to have minimal chromophore absorption in this range, which you can see here. So as we move out to the near infrared, we have minimal absorption. We have minimal scatter, so you can see that here. So the tissue chromophores are going down. And then we also have minimal autofluorescence. So as we increase in wavelength from sort of blue to red to near infrared, now we have a black background. And so we could specifically add targeted contrast here without having a lot of other fluorescence to compete with. And so because of all of these properties, we really have maximum photon penetration. So this will allow us to see structures that are buried beneath tissue that are specifically highlighted by exogenous contrast. Because of this, also the vast majority of the clinical fluorescence-guided surgery systems have been developed to image in the near infrared. So currently there's about 15,000 fluorescence-guided surgery systems that are in the operating rooms around the world. They largely are about 50% the Stryker system that I already talked about, and there's about 35% the intuitive system, and the rest are made up of some of these other systems that have a smaller sort of install base. So the question that my group has really been thinking about with this existing technology is if we could improve fluorescence-guided surgery outcomes for patients if we had nerve-specific probes that were tuned to the same color as the existing clinical fluorescence-guided infrastructure. And so the problem that we're really trying to solve is that nerves are typically pretty small, translucent structures. They're going to be buried, protected, sort of deep within the tissue, and it can be very challenging to identify them intraoperatively prior to when you actually get down to them and injure or transect them. So this is really difficult in general to repair damaged nerves, and so patients in general are left with pain, loss of function, and generally decreased quality of life. And so while neuroanatomy is, of course, well known throughout the body, by definition, patients that are having surgery have some sort of disease or injury, and so the neuroanatomy is going to be different than the neuroanatomy in the textbook. So here, just as an example, thinking about radical nerve-sparing prostatectomy. So here, this is the prostate. The vasculature is highlighted in red and blue, and then the nerves are nicely highlighted in yellow for differentiation, but of course the surgical reality is quite different from this. So one of my colleagues doing a radical nerve-sparing prostatectomy using the Da Vinci surgical robot sent me this image. So not being a surgeon, he also kindly annotated it for me. So this is the prostate, so this is this. And then this is the neurovascular bundle, basically illustrating here. And so what you can see is that they never actually see the nerves. They're just in the tissue pedicle where they expect them to be. And so our goal is to think about if we could make the intraoperative imaging look more similar to the neuroanatomical drawings. And so the accidental or iatrogenic nerve damage during surgery can occur throughout the body. It really depends on the surgical site and the proximity of the nerves. I want to show you a few examples of the types of images that we're able to generate using our contrast agents, and then I'll talk to you a little bit about how we got to this space. So here, just an example of looking at facial nerves. I'll show you all rodent images to start with, so we can see that we're able to see the larger facial nerves that you can see by white light are nicely highlighted by our contrast agents, but you can also see much smaller structures that are basically completely invisible by looking at the white light image alone. We can also look at nerve structures that are commonly injured during surgery. So this is the recurrent laryngeal nerve, runs next to your trachea, can be injured during thyroidectomy, causes voice change and voice loss for patients. You can also see the vagus nerve here. And then you can see here, we can also look at larger nerve structures. So this is the femoral nerve, not challenging necessarily to find in patients, but you can also see that we can see much smaller branches of the nerve. And so you can think about how you might utilize this technology in the operating room. So what my group has been doing for about the last 10-ish years or so is thinking about if we could make nerve-specific fluorophores like I just showed you, but be spectrally matched to the clinical imaging systems that have already been translated, so that we would just need to translate the probe alone and not do a probe imaging system coupled translation. And so I actually started this project when I was a postdoctoral fellow, and so what we were doing is working with this scaffold called the disterylbenzene scaffold. I would say that this contrast agent is nerve highlighting, not really nerve specific. And you can see that if you look at the brachial plexus nerve of the mice. You can see that this contrast agent is in the brachial plexus nerve, but there's lots of other stuff that is fluorescent too, so it's not really that specific. You can see the salivary glands, the connective tissue, the adipose. So this isn't especially helpful. Also, this is far from the near infrared. We have UV excitation and visible emission and heavy overlap with the hemoglobin spectrum, so it's going to be very challenging to use this intraoperatively. Around the same time, there was some nice published work out of the late Dr. Roger Chen's lab. So these are nerve-specific peptides. This is a targeting sequence that was found by phage display, and then a linker conjugated to a visible fluorophore. So they actually use Alexa fluorophore 88. And so you can see highlighting the brachial plexus nerve and the sciatic nerve. There's still some non-specific uptake and some in the adipose and here in the cut muscle tissue. But this technology has actually moved forward to clinical translation and is currently in clinical trials right now looking at head and neck nerves during head and neck cancer surgeries. And then this other similar strategy by Dr. Thomas Reiner's laboratory used a different nerve-specific peptide to highlight the nerves, and again used a linker and then a visible dye. So they're using a very similar colored dye. You can see that they're able to see the sciatic nerve, but there's a lot of non-specific sort of background fluorescence, likely due to the color spectrum that's being looked at. And then my friend and colleague, Dr. Hak-Soo Choi, published in 2014 that the oxazine scaffold, specifically oxazine 4, which is red but not yet in the near infrared, is quite nerve specific. And so you can see that if you compare this image to this image. So we're looking at sort of the same field of view, except for now we have high nerve specificity. We just see the brachial plexus nerves and not much else. And so what my group has done is what I would call sort of a fluorophore medicinal chemistry project, thinking about how we could take the oxazine scaffold and move it from the red out to the near infrared while still maintaining its nerve specificity. And so over the past 10-ish years or so, I guess we're getting closer to 12-ish, we've made a library of about 400 oxazine derivatives. And we kind of have some favored molecules that are in that near infrared space, both 700 and 800 nanometer in color. And the reason that this is important to us is sort of what I just said a few slides ago about the clinical infrastructure. So because of the existing 700 nanometer methylene blue and 800 nanometer indocyanine green, most of the imaging systems are designed around these two colors. In fact, the vast majority of the imaging systems are designed to image indocyanine green. So we're trying to work to develop both colored nerve agents because we're thinking about not only nerve visualization, but also the future of how fluorescence-guided surgery could be used. So, for example, you could use it for nerve and tumor imaging, for multicolor imaging. But of course, we're not the only ones who have been designing contrast agents, and so many of the cancer-targeted contrast agents are also designed to be visualized in the ICG channel because that's the most frequently used channel in the clinic. And so having a 700 nanometer agent will also help us to think about the future and using that multicolor strategy. Right. So how did we do this work? So we started out looking at these two molecules. So this is oxazine 4 that I showed you on the previous slide is highly nerve specific. You can see its excitation and emission is slightly below the near infrared, so below 650 nanometers. This is oxazine 1. So it's a close structural analog of oxazine 4. Its excitation and emission is above 650 nanometers. Now, even though these are very close structural analogs to one another, they have very different biodistributions. So changing the molecule slightly can have a big change on how the molecule accumulates in the body. So you can see here in mice, oxazine 4, highly nerve specific. Oxazine 1, really bright, but it stains everything. So there's basically no nerve contrast. We just have pan tissue fluorescence. So taking these two pieces of information, we made an initial library of about 65 compounds, and we ended up with some favored, might not be able to show you. Oh, here we go. Favored compounds up here that are both kind of in the near infrared and have high nerve specificity. So in the interest of time, I won't show you all the data. It's published, but what I will show you is that because of this, we are able to demonstrate that now that we've moved out into that near infrared space, we actually can visualize buried nerve tissue. So we did collaborative work using a modified Da Vinci surgical system. So we're looking at swine. So these are like 150-pound swine. We have the robot docked with their peritoneal space, and so we're looking at back at the body wall to where the lumbar nerves are running in the body wall. And so the tool is actually pointing to where there is a nerve, but it's completely invisible by white light. And in fact, we can only see it when we use the appropriate excitation and emission light. And so you can see it here. We did a number of these studies, and then we also did a bunch of what I would call fluorescence-guided dissection, because we wanted to verify that all of the linear things that we could see by fluorescence were actually nerves. And so we did cut out a bunch of these tissues and send them back to the university so that we could do histology and verify that, in fact, yes, these structures that we can see actually are nerves, and we're able to visualize them beneath about 3 to 4 millimeters of tissue in the native environment. So we were excited about this, but as I said, this is sort of in that 700 nanometer space. So of course, we would really like to have a molecule that's also in the 800 nanometer space, because it's going to be compatible with the vast majority of clinical imaging systems. And so we took kind of what we knew from the oxazine 1 and oxazine 4 space and some of our favored compounds that we produced in our initial library, and we did some kind of synthetic magic that I won't bore you with, since we're not in the chemistry department, we're in the engineering department. And so we were able to shift our molecules by 100 nanometers out into sort of that ICG-like channel. And so you can see that here, we got actually 100 nanometer redshift in our molecules. And so what this enabled is now we could actually use the existing Da Vinci Firefly without any modification to visualize the nerve tissue. So we're looking at the iliac nerve again in swine. What's also nice about this is because of the optical properties, you can see the nerve below the blood, even though there's blood in the field. And so we have really nice visualization. So we were really excited about having sort of both colors, 700 and 800 nanometer nerve dyes. And so what we wanted to move towards next is translation to the clinic so that we can actually use this in patients. And so Dr. Eric Henderson and I have been doing a collaborative project together, working on moving this forward. We actually have a multi-PI R1 grant in order to get the IND and then do the phase 1 clinical trial. So I can say those words really fast, but it's taken us many years to kind of move this forward, as I think is well understood by others in this room. And so as we've worked towards this project, we actually did a project with Eric and Logan, who just successfully defended his master's thesis yesterday, looking at deciding that we could actually see human nerve cross-reactivity of our probes. And so they came up with this really nice model where we could use freshly resected human limbs and a decompression perfusion pump so that then we could systemically administer our contrast agent to human specimens. And so I got lots of really cool early morning text messages after surgery when I'm like just getting up because it's 3 hours later on the West Coast with lots of winds and cool fluorescence, so very exciting. And what we found is that you could actually get really nice nerve-specific contrast in these models. So here you can see that the contrast is quite akin to what I just showed you in animal models. And then these tissues can be further resected, and we're able to nicely visualize that nerve-specific contrast is high, and we see little accumulation in the muscle and adipose tissue. So we're currently working together on our IND in order to move this forward to a phase 1 clinical trial. I am hopeful that our IND will be submitted within the next month or so. So we have been doing quite a bit of work to move forward with that. And then we'll be doing our phase 1 clinical trial at Dartmouth Hitchcock. So some other applications that I wanted to kind of touch on are some things that I've already said. So again, we are excited about the idea of doing 2-color fluorescence-guided surgery. So in collaboration with Kim, we've also been looking at mouse models of different cancers and how we might be able to use our molecule that we're translating already through an IND with ABY029. So here, this is an example of a sarcoma tumor implanted next to the sciatic nerve. So the sciatic nerve there within the arrowhead, the sarcoma here kind of infiltrating into the sciatic nerve, and then co-administration of these 2 spectrally distinct molecules. And so you can see we get really nice distinct highlighting of the nerve tissue and the sarcoma tissue, which you can see here, and we can make this really nice merged image where you can visualize both of these structures. Here, another example using the same 2 dyes, but now we implanted a head and neck tumor next to the facial nerves. And so you can see, again, that we're nicely able to separate spectrally those 2 structures, and we actually see a dark spot on the nerve where the tumor is overlaid, and we can see this nicely using the ABY029 contrast agent. And so we're starting to think about how we'll actually move this to the clinic. Will we need like separate INDs for the 2 agents? How will we actually do the administrative part of co-administering the fluorophores together? What will that look like in the eyes of the FDA? And what kind of 2-color imaging system will we use? There isn't currently such a thing that exists that's clinically available. Some other nice applications that we started to look at is we're actually quite interested in the idea of nerve-sparing radical prostatectomy. So the Da Vinci I showed you has a really nice channel for visualization, and so we could have the opportunity to visualize the prostatic nerves during routine radical prostatectomy. About 90% of prostatectomies are actually performed using the Da Vinci surgical robot. So we were able to demonstrate in canine prostate that we can really nicely see the prostatic nerves. So you can see the nerves around the prostate and actually on the prostate. So here this is the canine prostate, and so we can see the nerve tissue kind of on the prostate, behind the prostate, and then above the prostate. We were able to resect out these tissues and again send them back to the university so that we could look at the tissues by conventional pathology, so H&E staining. We found that we could see very small innervation on the outside of the prostate and then within the prostate, and we also were able to verify that in fact these are nerves using conventional antibody staining, so you can see positivity in those spaces for neurofilament staining. Rather recently, we actually learned that it is actually untrue that all of these contrast agents are specific for the peripheral nervous system, and in fact there is a subset of the molecules in the library that we produce that can cross into the central nervous system. And so this was surprising. I actually had an MD PhD student that rotated in my lab that was really interested in neurosurgery and wanted to try this out, and so our discovery this day, we thought, oh, I'm really sorry, we stopped screening all the molecules for their CNS specificity and stopped looking, so now we need to go back and look again. And so now we kind of have a thought about what portion of the library is specific for the central nervous system. We've been thinking about some different applications here. So one application is to look at skull-based surgeries where we can see the relatively small cranial nerves at the base of the skull. So here illustrated anatomically, and then tumors can kind of grow intimately associated with these nerves. And so you can see here what the surgical reality looks like. So in surgery, surgeons have to spend quite a long time separating the tumor from the nerves, and it's very challenging to visualize the 2 structures together. So we're working on this project in collaboration with Dr. Linton Evans. And we have been developing these contrast agents that are bright in the central nervous system and allow us to see the optic nerve and the really small cranial nerves at the skull base, which you can see here. And so we have kind of some favored agents that we're looking at in collaboration with Dr. Scott Davis and Augie and Caleb. They've helped us to look at three-dimensional rendering of some of these contrast agents. So this is one of our favored agents that's able to cross into the central nervous system. And so you can see some really beautiful cryomacrotome images showing the trigeminal ganglia, the brain, the spine. We can also see the peripheral nerves. So here is the brachial plexus nerve. And we then wanted to compare this to if we just look in the autofluorescence of the tissue, so we can see we have quite nice specificity of our contrast agent for the central nervous system. And then another application that we started to look into after we discovered this central nervous system specificity is that we thought, well, I wonder if some of the molecules can actually cross into the brain. Maybe they'll cross the blood-brain barrier. And so the application of interest here is to help during glioma, glioblastoma surgery for looking at the white matter tract, so looking at the functionality of tissue. So diffusion tensor imaging, diffusion tensor MRI is commonly used to map the white matter tracts prior to surgical intervention, but then intraoperatively, the tissue moves significantly when you open the cranium, and so electrophysiological monitoring is used, but this is just point-based measurements, not really imaging. And so then, again, the surgical reality is quite different from what was done prior to surgery and the type of imaging that was conducted. And so we've been looking at using our fluorescence imaging agents that can actually cross the blood-brain barrier and highlight the white matter tracts to help to augment the preoperative imaging. And so we're working on a collaborative project right now thinking about could we make spectrally distinct probes and use maybe the ALA PP9 system and then sort of the 800 nanometer channel to visualize the white matter tracts. And then I want to just talk about one other quick project that we're working on in this space before I switch topics and talk to you a little bit about our project looking at drug distribution and binding. So kind of the last thing that we've been working on in this space is thinking about visualization of nerve health and whether our fluorescence imaging can actually be functional. So like functional fluorescence imaging, like functional MRI. So we also discovered kind of serendipitously while we were teaching new people to do nerve exposure surgeries, that if nerves are damaged during those exposure surgeries, we actually see a change in contrast. And so here, looking at an example both in mice and rats where we've done a purposeful sciatic nerve crush, you can see that we're able to visualize it in the white light channel, but if we look in the fluorescence channel, you can see a big fluorescent contrast difference. So this is a sciatic nerve injury, and then we administered the contrast agent to the rodents. You can also see, if we look biconfocal, fresh tissue confocal, so you can see a big change in the fluorescence in the injured area and a lot of fluorescence in the healthy, uninjured area. We also looked at the idea not only of acute injury, but also the idea of iatrogenic or accidental surgical injury. So here, the difference is that the contrast agent is already on board, and then we're looking at what happens if the injury occurs during surgery. So we actually did the crush once the contrast agent was there, and we could actually watch the fluorescence decrease over time. So this is 15 minutes, 30 minutes, an hour after the injury occurred, and so we can actually see that contrast going away. So you can imagine maybe using this at the end of the surgery to look at nerve integrity. So the project that we've been doing is looking at whether or not we can use the early fluorescence changes after injury to predict whether the injury is permanent or recoverable. And so this is a project of one of my graduate students, still ongoing, so the answer is unknown, but I will show you the data. So we've been looking at different models of nerve injury, trying to make a robust injury that's both permanent and recoverable, which turns out to be hard in rodents. And then we are trying to compare that to standard electrophysiology monitoring, our in vivo imaging, and then gold standard histopathology of our harvested nerves. And then I'm going to show you a time series of data. So the way that we did this experiment is we did a sterile nerve crush injury at time 0, and then we closed the animals back up. And then we looked imaging short time points and long time points. In every one of the images that I'll show you, we administered the contrast agent an hour before we did the imaging. And this is actually the contrast agent that we're currently working on the IND for. And so what we found is quite interesting. So in all of these images that I'm going to show you, the spine of the mouse is here, the foot is here. So this is proximal to the injury, and this is distal to the injury. I'm just going to show you one representative image from each cohort of the injured side. We also looked at the contralateral side for control. I'm going to show you the white light image, the fluorescence image, and then the 3D quantification of the fluorescence image. So here we're looking at transection, so cutting out a piece of the nerve, and then the crush injury. And so what we found is that you can already kind of see the pattern a little bit. So one day after injury, proximally to the injury, we have decreased fluorescence intensity, and distally to the injury, we have increased fluorescence intensity. It's not as obvious in the crush, which is a less severe injury, but in the short time points, you can actually see this pattern appear if you look 3 days, 5 days a week out. So lower fluorescence proximally, higher fluorescence distally. And then at 2 weeks, the fluorescence intensity is equalized across the nerves. You can really see that here in the crush injury. You actually can't even see a decreased fluorescence where the crush is anymore. You see the space where the nerve is cut out, but the fluorescence on each side is equivalent. And then at 3 weeks, the pattern actually reverses. So now you can see increased fluorescence proximally and decreased fluorescence distally. And this is maintained for the remainder of the experiment. So still to be determined, and we're looking at other like lesser models of injury, but we have this kind of interesting pattern, and we're trying to kind of work together with Eric and Dr. Jennifer Hong to try to understand sort of what this means contextually. Okay, so I'm going to stop here with this project, and I'm going to switch gears and talk about the other fluorescence imaging project that we've been doing development on in my lab. And so for motivational purposes, I want to just talk for a minute about kind of the problem that we're trying to get at. So we've been thinking a lot about why targeted therapeutics don't cure advanced cancers. And the real reason, I think, is that there's really high heterogeneity, and it's really hard to tell if your drugs are actually hitting the target. And so I think that we have something to offer in this space, and I'm going to share with you how we've been using the paired agent imaging idea to label drugs that can label their drug targets intracellularly. And so the questions that we're trying to answer are looking at what's the local drug concentration, how many of the drug targets are actually engaged after therapy, and then we've coupled the paired agent imaging idea to cyclic immunofluorescence, which I'll talk about in just a second, to look at how the spatial proteome is affected by targeted therapy. And so in collaboration with Kim and Ken, we've developed this platform technology that's the 3 imaging technologies together that we call TRIPOD. So this stands for Therapeutic Response Imaging through Proteomics and Optical Drug Distribution. And what we're currently using it for is to look at tumor heterogeneity in xenografts, as well as therapeutic response and resistance. And to date, for proof of concept, we've been really focused on the idea of looking at tyrosine kinase inhibitors. So I'll talk to you about each of these technologies in just a second, but basically we're making fluorescently labeled tyrosine kinase inhibitors. So they're here, this is the EGFR targeted erlotinib. And then we're using the paired agent imaging idea, except for we're using cell membrane permeable drugs. So now we have the opportunity to look at intracellular drug targets and be able to quantify how much drug is bound using what Kim usually talks about as the binding potential, but we've kind of relabeled it here and called it the drug target availability. And then we've coupled that with our cyclic immunostaining strategy to actually be able to look at the spatial proteome. And then I'll show you that we've done some proof of concept studies where we've coupled all of this data together and looked at the single cell level to try to get sort of a mechanistic understanding of drug response. So I want to talk about how each of these technologies works and then show you the data of all of this together. So potentially some of you might be familiar with this idea of cyclic immunofluorescence. So this is really the solution to solve the problem that usually microscopes are really spectrally limited. So your microscope can probably image in 3 colors, maybe 5 if you're super fancy and using all of the spectral space. But that's not going to help us to visualize the proteome, which is made up of many tens to hundreds of proteins that we might be interested in. And so The solution that many people use, including ourselves, and there's lots of technology out there to do this, is that we use this idea of cyclic staining. So we basically stain, image, erase, and then go back and stain, image, erase again. We take multiple images and then build up our imaging data cube by doing this cyclic process. So the way that we do our stain, image, erase is we use DNA bar-coded antibodies. We use a single-stranded oligonucleotide we call a docking strand to label the primary antibody. Then we label our samples with the DNA bar-coded antibodies, and we can bring in our fluorophore labeled imaging strand, which will hybridize in situ to the docking strand on the primary antibody, and we can image this just like any common indirect or direct immunofluorescence. So like this. Then the erase bit is that we use a photocleavable linker. So here, our PCL photocleavable linker. This is UV labile. So when we're done doing our imaging, we shine UV light onto the sample, and then the fluorophores are clipped off, and we just have the oligonucleotides closed in this space. And so then you can imagine how this could work then is that we were able to label our samples. So here I'm just showing you 6 antibodies. They all have different docking strands on them. We can bring in 3 imaging strands because we don't have a fancy microscope, so we're just going to image 3 colors at a time. So those will hybridize specifically to their docking strand that's complementary, and then we can image, treat with UV light. All of the fluorophores removed, and then we can come in with imaging strands labeled with the same 3 fluorophores, but now to different targets, and we're able to, again, image the sample. We can clip these off. We could continue along. And so we have published this methodology. So here, just looking at it in a breast cancer resection sample where we've done 4 cycles of 3-color imaging, so looking at different targets, and then if you zoom in, hopefully you can see. Oh, it's really hard to see up here. Apologies. If you zoom in, then you can actually visualize the individual staining patterns. So we can look at different cytokeratins, we can look at proliferation, we can look at the immune cells, different structural markers. And so this is the technology that we want to couple to look at the spatial proteome with our paired agent, intracellular paired agent imaging technology. Potentially some of you are familiar with this idea, so this is not a new idea. The idea of paired agent imaging was actually developed first in the 50s, so it's from the nuclear medicine world, and the challenge that was recognized is if you use a radio-labeled antibody and you try to image the tumor, that the amount of accumulated antibody isn't necessarily related to the amount of target that's there. And that's because of the enhanced permeability and retention effect and the fact that you just get a lot of non-specific accumulation in the tumor. And so the solution to this was to use 2 antibodies that are radio-labeled with different with isotopes of different energy, and then to take a dual energy image and take the ratio in order to quantify the amount that's actually labeled. And so possible in nuclear medicine, but easier in the fluorescence space because we can readily do multicolor imaging. And so I borrowed from Kim's literature some really nice work showing that this will actually enable you to visualize the EGFR content in tumors. So here, this is some work showing tumors that have varied level of EGFR from high to low. The tumor is here in white. But you don't necessarily see fluorescence intensity related to the EGFR expression. However, if you use an antibody labeled an EGFR antibody and then an antibody labeled in a different color that's not specific for anything, and you take dual color images and then ratio the 2, so you just take the targeted image, subtract the untargeted image, and divide, then you can get these paired agent images where the intensity is strongly correlated to the amount of target present. And so I would say that this idea has been reinvigorated by the optics community. So it got first published in 1957, published really nicely by Kim in 2014, and then published by the groups here and ourselves, showing that this technology can be really useful in the optical imaging space. And so our take on this has been to move to using small molecules so that now we not only have access to the extracellular proteins that are on the membrane that you can look at by antibodies in live cells and tissues, but to use small molecules that can actually cross the membrane like drugs so that we have the opportunity to look at the intracellular proteome and also at drug targets. And so we have initially done this project for proof of concept, looking at EGFR tyrosine kinase inhibitors, because we're able to look at the intracellular domain using our technology, and then the extracellular domain using conventional antibody technology for validation. And so we're going to look basically at the tyrosine kinase binding. And so what we're going to see then is hopefully how many of the drugs are actually bound at the target site to cause actual therapeutic effect. And we're going to look then at the drug target availability. And we could look at this both in the presence of parent drug treatment to find out how many sites are actually open after the parent drug has been dosed, or we can look at it natively just to quantify the number of potential drug targets and thinking about optimal dosing for patients. Yeah, so I said those things. Yeah, okay. So then what we did in order to label our molecule is now we need to make the targeted and untargeted version of a small molecule. So we're using the small molecule orlatinib. Here it is docked into its protein site here. And so you can see, if you look at the molecule, this is the part of the molecule that is really important for binding. These are the solubility tails, so less important for binding, more important for biological compatibility and giving the drug to patients. And so what we've done then is choose to do our targeted labeling out here, away from the binding site, and do our untargeted labeling here. And we've used two molecules initially, tetramethylrhodamine, so TMR, and silicon tetramethylrhodamine. So the only structural difference is here. Switching the oxygen for a silicon atom actually shifts the color of this molecule by 100 nanometers. But they're structurally very similar, so when you bind them to the small molecule, they should have relatively similar biodistribution in cells and tissues. So we learned something kind of interesting along the way, initially frustrating, but then we discovered sort of the challenge. So we were having a lot of variability in our intracellular paired agent imaging when we're using TMR, silicon TMR. And so the reason behind this is actually that these molecules can isomerize, meaning that they can change from this open carboxylic acid version to this closed version here. So this molecule is actually non-fluorescent, and so this happens in different environments. So actually it's published in the literature that people use this in order to have a fluorescent sensor to look at environmental differences in cells and tissues. It's great for that. It's really bad for ratiometric imaging when you're trying to use the fluorescence intensity to tell how much target is actually there. And so on the side, we did this other project that we published last year, showing that we could redesign these molecules in order to get away from that isomerization and closed form non-fluorescent version. And so initially what we did is just take away the carboxylic acid, and we made this molecule. The problem with this molecule is that it's really sticky because it's positively charged. So it has really high background across the cells. It sticks to all of the intracellular organelles. And so instead, what we decided to do is we added this sulfonate group. So this balances out the charge. So now we have a neutrally charged molecule. It still can freely penetrate the cell membrane. The sulfonate gives it really nice water solubility, and then it also has low background because of this litter-ionic nature. And then most important to us, it's always on, so we don't have that environmental sensitivity. So then we ended up using these new dyes that we call the orthopores that we developed. So same molecules, basically same color. So we have 100 nanometer redshift between OF550 and OF650, and now these are the molecules that we used to label our small molecule erlotinib. So I'll show you in just a second. We made all the different colors of this. So we made the targeted version labeled with OF650 and OF550, and the untargeted version labeled with the same two dyes. For experiments, we ended up using this combination of molecules because we have lower concentration of accumulation of our untargeted drug than our targeted drug, and 550 is brighter than 650, so that's how we ended up using it for imaging. But for validation, we looked at all the different combinations. Now you might be thinking, this is a small drug molecule, and I am making a big change to the drug molecule. Is the drug molecule still going to bind at the site of interest? And so we have actually done quite a bit of work to look both modeling and in vitro, which I won't run through all the studies, but to demonstrate that we actually do still have competition of the targeted molecule with the parent drug. So this is modeling studies looking at the docking score. So our targeted molecule actually has quite similar behavior to the parent drug, and our untargeted molecule has much lower docking score, meaning that it's not binding at the site. And so what we actually care about most here is that we have some competition with the parent and that we have a big differential between the targeted and the untargeted. We really don't want the untargeted to interact at all with the drug target, so we can actually quantify how much drug is bound. And then just for one more verification here that this actually works, we did do in vitro staining, so this is live cell staining. So we're staining with extracellular EGFR antibody. You get really nice membrane staining in EGFR positive cells. And then this is the targeted version of the molecule, so we did make it in both colors. You can see that we get really similar staining pattern. And you can also see that we have really nice overlap of the three. So this verifies that, yes, OF550 and OF650 have very similar behavior, at least in culture. And they do, in fact, allow for erlotinib to still stain the membrane. By comparison, if you look at the untargeted molecule, so all we've done is just switch where the label is on the molecule so that it will now be in the binding site. So we've done the same experiment, so EGFR antibody staining, and now staining with OF650 in the untargeted way to erlotinib and OF550 in the untargeted covalently labeled to erlotinib, and we basically have no membrane staining, so we've taken away all of that specific behavior. All right, so now what are we going to do with this? So initially, we looked at just some optimization experiments. We did quite a bit of work looking at the pharmacokinetics of these agents, the dosing, how we might use them to look at distribution in mice. I won't show you all that, but to say that it is published work, but instead I want to show you that we've actually looked at treating with the parent drug and then doing the last dose with the IPAI fluorescent agents in order to look at parent drug distribution, response, and resistance. So this is the experimental setup. We xenographed our tumors into our mice. We orally administered them the parent erlotinib, not fluorescently labeled or changed in any way. This is the way it's given in clinics. So we gave a BSA scale dose to the mice. And then we IV injected our probes just before sacrifice, and then we resected and froze the tissues, sectioned them on slides. We did whole slide scanning and tiling to look at the entirety of the tumor. So we looked first just at the IPAI agents, so distribution. These aren't covalently bound in the tumor, though, so we can easily saline wash and get the tumor to basically come back down to the level of autofluorescence. All of that is removed. And then we can come back in and do our cyclic immunostaining using our DNA barcoded antibodies. We can, again, scan and tile the entirety of the tissue together. And then after we have all of that data registered, we can single cell segment and look at spatial distribution patterns of the drug and the response and resistance by cyclic immunofluorescence. Oh, there we go. Sorry. Okay. Sorry, slides got ahead of me. All right. So in this initial study, one of the things we did is we wanted to look to see how long after treatment should we give the IPAI reagents. And so I'm showing you we did just a single oral gavage dose of the parent drug, and then we looked at an untreated group, a group that we gave our IPAI reagents 6 hours after, 12 hours after, and 24 hours after the parent drug dose. And so then here I'm just showing you violin plots for EGFR and phospho-EGFR, and the takeaway point is you don't really see any difference. Potentially you shouldn't, because we only gave a single dose of a therapeutic that's dosed daily. But what we can see is that we actually see a difference in the drug target availability. So at 12 hours, we actually see a decrease in the mean drug target availability, meaning that the parent drug is occupying those sites. So we see a lower ratiometric drug target availability that returns to normal within 24 hours. Also, potentially, what we expect to see, because again, this drug is dosed every 24 hours, because that's its pharmacokinetic profile. And so the other thing that we did here is really look at the individual tissues. And so I kind of want to show you what we can see. So here, I'm going to show you different panels. The top panel is going to be three-dimensionally. So the drug, the DTA, the drug target availability, is in green, and it's also height mapped. So the height mapping is also the fluorescence intensity. And then we're looking at potentially the other important markers. So we're looking at phospho-EGFR, so the phosphorylated state of the protein that's being treated by erlotinib. We're looking at proliferation in blue by Ki67. We're looking at apoptosis in yellow, so dead cells by cleaved caspase 3, and then you can see the individual nuclei in white with DAPI. And so I just want to show you the kind of changes that happen over time. If you're interested in the other markers we stain for, they're two-dimensionally down here. So you can see sort of here, already at 6 hours, we see quite a big change. So we had sort of heterogeneous, mountainous drug target availability without treatment. And 6 hours after treatment, we have quite a decrease in the drug target availability. So many of those drug targets are actually bound. But we also see that we still have strong proliferation. We see a lot of Ki67 signal, and we don't see a lot of dying cells, cleaved caspase 3, maybe a few. But if we look 12 and 24 hours after treatment, we see quite a different distribution. So you can see that now we're starting to see dying cells. We see quite a bit of yellow. We still see that decrease in drug target availability and not as many blue proliferating cells. And then at 24 hours, while we still see some dying cells, we also see proliferation kind of returning here. And so you can really see this stark difference if you compare the untreated animals to the 24-hour treatment animals. Now, we've also done longer treatment studies, and so this is continual oral gavage dosing with erlotinib, and then looking at the drug target availability. So here I'm just showing you drug target availability and EGFR staining. So this is after the tumor either shrank or grew, and we decided to resect and look at it. You can also look at this in the same way, three-dimensionally, so we definitely have these islands of cells that have high drug target availability, meaning the drug is for some reason not reaching them, and those cells are really not being treated. And then we have these other cells where we don't have high drug target availability, and so those are presumably having good treatment efficacy. So we're still working on finishing up this study, but we've done this sort of platform development, and now we're looking to extend this technology to look at other cancer types and cancer treatments. So I am going to stop here and just conclude with a few things, and then I'd be happy to answer any questions. So I hope that I have convinced you that near-infrared contrast agents could change clinical medicine. And so I've talked to you about how fluorescence-guided surgery can improve intraoperative accuracy, but we need near-infrared targeted contrast agents. My group has been working on making near-infrared nerve-specific contrast agents, specifically 700 and 800 nanometer candidates. I've shared with you our idea about just nerve identification and visualization. I'm thinking about multicolor fluorescence-guided surgery applications, how we think we might be able to use this in the central nervous system space, both for cranial nerve imaging and for white matter tracts. And then our idea about imaging nerve health and thinking about functional fluorescence imaging. And then I shared with you our other sort of more preclinical project, thinking about personalized cancer medicine and how we need to really understand the relationship between the tumor microenvironment, the tumor cells, and the drug distribution to make personalized medicine for patients. So we've been developing this tripod imaging platform. Along the way, we did this novel fluorophore development work, and now we plan to expand this technology to other therapeutics and cancer types. So with that, I would like to say a big thank you to my group, to my collaborators, both here and at other universities, and of course to our funding. Thank you for listening. I would be happy to answer any questions.